Hello, everyone. It's good to see you today. I'm happy to be with you. My name is Steve Holland, and I am the Vice Chair of the National MPS Society and the father of 3 MPS I children. And it's my pleasure to moderate for you today our last session of the day, which is session number 2, Newborn Screening, Pioneering a Second Nomination. We're very blessed to have 3 phenomenal speakers with us this afternoon, the first of which I am going to introduce now. But before I do that, I do want to point out that at the end of the 3 speakers, we're going to have a Q&A session. So I would ask you to go ahead, and as you have questions, please go ahead and click on the button that says Ask a Question and get that written down so that we can have that when that session goes live and we can answer your questions. Our first speaker is Barbara Burton, MD. Dr. Burton is a professor of pediatrics at Northwestern University and clinical practice director and director of the MPS-ML treatment program at Children's Hospital of Chicago. She's a pediatrician and geneticist whose research interests are focused on inborn errors of metabolism and newborn screening. Dr. Burton is an investigator in numerous clinical trials, including MPS, with over 200 peer-reviewed articles. She served for 4 years on the Federal Advisory Committee that makes recommendations regarding newborn screening in the United States, and currently serves as chairman of the Newborn Screening Advisory Committee for the state of Illinois. We welcome Dr. Burton, who will speak on newborn screening. What is it? How will it help the diagnostic journey?
Hello, it's such a pleasure to be joining you today for the annual conference of the National MPS Society, and to have the opportunity to talk about one of my favorite topics, which is newborn screening. So our title today is Newborn Screening, What is it, and how will it help the diagnostic journey for the MPS disorders? Here you see my disclosures. I do some consulting and clinical trials funded by pharmaceutical companies. And so first, let's just start by talking about what is newborn screening. I think those of you in the audience who've had a baby probably remember that there was a blood test that was done from a heel stick when the baby was typically 1 to 2 days of age. And the blood from that heel stick is spotted onto filter paper like you see on the screen. This is done in every hospital in every state throughout the United States, and is also done throughout the developed world. And many people refer to this newborn screening test as the PKU test, but in reality, newborn screening in every state includes over 35 different disorders, and more are being added all the time. Newborn screening in the U.S. is what we would refer to as a state health department initiative. The testing is typically done in state health department labs, so samples are sent from birth hospitals or from home births to the state laboratory where the testing is performed. There are a few states where states band together and send testing from one state to another. But each state decides which disorders will be included in the newborn screening panel, and there is some variability from state to state. This variability has decreased somewhat over the years since a federal advisory committee was created called the Secretary's Advisory Committee on Heritable Disorders and Genetic Diseases in Infants and Children. This is a committee that makes recommendations to the Secretary of Health and Human Services regarding which disorders should be included on what is called the Recommended Uniform Screening Panel, or RUSP. This is a list of disorders that the committee and the Department of Health and Human Services have said should really be included on the newborn screening panel. But this is a recommendation to the state. It is not mandatory that states follow the recommendations or that they screen for all of the conditions on the RUSP, although most states eventually do adopt the conditions that are included. So to really get widespread newborn screening for a particular condition, it is very important to have that disorder listed on this recommended uniform screening panel. Some states may choose to include disorders that are not on the RUSP, and this has certainly happened many times in many states, and this can be done in some cases just by administrative rule changes or decision-making within the Department of Health or through legislation. So there are currently 35 different conditions on the RUSP, including one MPS disorder. That is MPS 1, which was added in 2016. There is also an established mechanism for nominating new disorders, and nominations can be made by individuals, by advocacy groups, or by healthcare professionals. MPS 2 was nominated this year by the National MPS Society, and the nomination was recently forwarded to what is called full evidence review. In other words, the advisory committee decided there was enough evidence that addition of MPS 2 would be of value that it should be studied intensively by a group who conduct what is called the evidence review and then come back to the committee with their recommendations and with their findings. This process can take up to a year, so adding a disorder to the RUSP is not a quick process, and while we're very hopeful about MPS 2, there is no guarantee at this point that a positive vote will occur at the end of this process. Although certainly I hope very much that this will be the case. In general, there are a number of criteria that must be fulfilled to make a disorder a good candidate for newborn screening. First of all, of course, the disorder should be a serious one. There has to be a test that reliably detects the condition on a sample from the dried blood spot without an unacceptable number of false positive test results that require babies to come back for more and more testing. There should be treatment available, and there should be evidence that initiation of treatment prior to the time that a clinical diagnosis would be reached is beneficial. In other words, if you can diagnose a child with a condition just based on what you see on exam, and if you start treatment at that point and everything is fine, then you really don't need newborn screening. So we are targeting conditions where it makes a difference if treatment is started before the time that doctors would normally make a diagnosis. There also need to be some pilot data from pilot screening programs showing that screening can be accomplished. Not only there is a test, but that test works in practice and the disorder can actually be detected. And our Federal Advisory Committee likes to see some pilot data from the United States. Although screening goes on in other parts of the world and the data can be reviewed and assessed from screening programs elsewhere, they really do prefer that there be some experience in the United States. So why would we consider newborn screening for the MPS disorders? Well, one very obvious reason is that it allows families to avoid the diagnostic odyssey that's experienced by so many. In many of the patients I see with MPS disorders, there have been symptoms for some time, and patients may have been seen for three or four or five different physicians in different specialties for different symptoms that relate to the disorder, but it really hasn't all been put together with a unifying diagnosis for some time. And that delay can be months, or in some cases, it can even be years. Families know there's something wrong, but oftentimes it takes a while to get to an accurate diagnosis. And that's a very difficult time period for everyone involved. In addition, we know that with the MPS disorders, the early initiation of treatment before permanent irreversible damage occurs is very important. With virtually all the signs and symptoms we see, it is easier to prevent them from developing than it is to reverse them once they are established. So we know early treatment very much improves the outcome for our patients with MPS disorders. And finally, if we make a diagnosis by newborn screening, we're in a better position to provide genetic counseling to families with information about their reproductive options for future pregnancies before they have other children. Sometimes a child will be diagnosed with an MPS disorder, and already there are 2 or 3 younger siblings in the home as well, and some of them may also be affected, and that's a devastating thing for families. So having that information in advance allows families to plan and make the decisions that are appropriate for them. So the MPS disorders really meet all of the criteria to be good candidates for newborn screening. And when I say the MPS disorders, I really mean all of the MPS disorders, not just MPS 1 and MPS 2. The disorders are certainly serious. We do have tests that can be performed for most, if not all of them, on the dry blood spot samples. We know that treatment is available for many for MPS 1, 2, 4A, 6, and 7, and there are investigational therapies being explored for MPS 3 as well. So there is evidence, in addition, that treatment of the disorders prior to clinical recognition is beneficial. This is true certainly with MPS 1. We know that the earlier transplant is performed in patients with the severe form of MPS 1, the better the outcome. With MPS 2, and we have anecdotal evidence in families also with just MPS 6 and MPS 4, other MPS disorders, that if we start a younger sibling very early on therapy, we see much less in the way of the clinical manifestations than we see in the older child who was diagnosed clinically. So early treatment is really very important. Pilot data from the state of Missouri were the basis for the addition of MPS 1 to the RUSP. And we now have pilot data from Illinois and Missouri, which are supporting the application for MPS 2. So you might think, well, how did these states get that pilot data if these disorders weren't on the RUSP when they obtained it? And they got it because there were families in those 2 states that went to their legislators and said that we want newborn screening for MPS 1, MPS 2. We want it because we have children who are affected, and we know that early treatment makes a big difference in the outcome. And so those states started screening for those disorders, even though the conditions were not yet on the RUSP. So newborn screening for MPS 1, because it is now on the RUSP and has been since 2016, is becoming widespread in the US. It's also ongoing in Taiwan. And there are 3 different methods that can be utilized, all of which work well, but all of which are based on enzyme activity. So the way screening is done is by looking at the activity of the enzyme that's affected in MPS 1, that's alpha-L iduronidase. And if that activity is low in the dried blood spot, then additional testing is done to confirm the diagnosis. If the technology is improved, it may one day be possible to actually measure glycosaminoglycans, the substances that accumulate in the MPS disorders, as a primary screen on that dried blood spot. And that would potentially allow us to detect all of the types of MPS with that single test, although we then have to differentiate among them. Right now, the technology is just not good enough to use the GAG testing as a primary screen, but it is used in some cases as what we call a second-tier test, meaning that after you measure the enzyme activity, then the GAGs are measured in the dried blood spot, and the test is called positive only if the GAGs are elevated. Some states are utilizing this. Other states are referring babies just as a result of the low enzyme activity for further testing by the consulting physicians. Now, this map shows you what I believe to be pretty current in terms of which states are doing screening for MPS 1 and MPS 2. The states colored in purple are doing MPS 1 screening only. I think it's pretty up to date, but there could be one or two states that are actually screening that I haven't colored in. I tried to visit as many state Department of Health websites as possible, but they may not always be updated, you know, in a timely fashion. The two states shown in blue, that's Missouri and Illinois, are screening for both MPS 1 and MPS 2. I would say that if this is an issue of interest to you and your state is not colored in, I would advise you to go back and contact someone in the newborn screening branch of your state health department and find out if they are in the process of implementing MPS 1 screening at a minimum. And if they're not, then why not? And be proactive in trying to get this really out to every baby in our country. This has been on the RUSP now for 5 years, and it's really time for this to become something that is available to every baby out there. Right now, I think about 2/3 of our newborns are having screening for MPS 1, which is a tremendous achievement, but we really want it for everyone. So if you have a positive screening test for MPS 1, what happens next? Well, first and foremost, a positive screening test does not mean that the baby has MPS 1. It means that further testing is necessary. That's what a screening test is. It selects out patients who need additional testing. It doesn't really make a diagnosis. And there are some other reasons why that screening test, that initial measurement of the enzyme activity, might be low. One reason is pseudodeficiency for the enzyme, which I'm going to tell you about in a minute because it is very important. Or sometimes the baby is a carrier of MPS 1, and carriers can have low enzyme activity. But that can be sorted out with the further testing that is done. If a baby is diagnosed with MPS 1, then the next step is to determine if it's the severe or Hurler form of MPS 1 or attenuated MPS 1, because that will influence the treatment choice, what the appropriate treatment would be for that child. And this we can usually do with molecular or DNA testing. But if not, over time, we can sort this out on a clinical basis. So I mentioned pseudodeficiency as one reason for a positive screening test. So let me tell you a little bit about what pseudodeficiency for MPS 1 is. And I'll also tell you that we see it really for all of the other lysosomal disorders as well. And I think we're going to see it probably for all of the MPS disorders once we start newborn screening for the others. We certainly see it for MPS 2. So pseudodeficiency is a situation where there's a variant in the gene. In the case of MPS 1, it's the iduronidase gene. So there's a change in the gene, and that gene change leads to very low enzyme activity as measured in the laboratory from that dried blood spot or from a whole blood sample. And in some cases, the activity can be as low as we see in true MPS 1. But in the body, there's enough enzyme activity to prevent any accumulation of GAGs, of glycosaminoglycans, and to prevent any development of disease. So patients with pseudodeficiency never develop any findings of MPS 1. It's a situation where it looks like the patient has an enzyme deficiency because we're measuring it in an artificial setting in a test tube in the lab, but in the patient, there's enough activity, enough enzyme activity to prevent the development of any clinical findings whatsoever. Pseudodeficiency for MPS I or for iduronidase is very common in a newborn screening population. I can tell you that in the state of Illinois, we find about 100 babies with pseudodeficiency for every one baby with true MPS I. So it's common, and it's particularly common in African Americans. So if you're in a state with a large African American population, you'll see a lot of pseudodeficiency, whereas, you know, it is much less common in other ethnic and racial groups, but it does occur in all groups. And we know that in pseudodeficiency, both the urine gags and the dried blood spot gags are completely normal because there's enough enzyme activity in the body that the patients don't accumulate that enzyme substrate, the glycosaminoglycans, at all. And so that's how we sort it out. We also know what some of those variants are in the genes. So if we do DNA or molecular testing, that also helps confirm that we're dealing with a case of pseudodeficiency and not MPS I. So if you have a baby who tests positive for MPS I, but the urine gags are normal, treatment like hematopoietic stem cell transplantation, which we use for Hurler syndrome, the severe form of MPS I, should never, ever be considered. It's not even on the table. And invasive testing like X-rays and MRI are not indicated. This is a situation where this is most likely pseudodeficiency. So you want then to get the genotyping, the DNA, and if the DNA testing, the genotype shows known pseudodeficiency alleles, in other words, changes in the gene that we know are well described to cause pseudodeficiency, not disease, then no further follow-up of that baby is needed. That's a normal, healthy baby. We do not need to do any other testing on that baby, and we don't need to continue to watch it over time. Now, if the genotype is inconclusive, then in some cases, repeat evaluation over time and repeat testing of the glycosaminoglycans may be appropriate. We think that virtually all patients with MPS I will have elevated urine gags, even at birth, but there's a little bit of doubt about whether that will always be true for the patients with attenuated disease. And that's why some physicians would choose to continue to follow those patients over time and just assure themselves that the urine gags remain normal. Now, I mentioned before that dried blood spot gags are used in newborn screening in some states with second-tier testing. The state of Kentucky is one. Their testing is actually done by Mayo. And it is done initially by enzyme determination, but if the enzyme activity is low, the gags are measured in the dried blood spots, and the screen is only called positive if the dried blood spot gags are elevated. In over 130,000 infants, they had only 2 positive screens, and one of those was truly affected, and one was a carrier. So using dried blood spot gags as a second-tier test does really get rid of all of the pseudo-deficiencies, other false positives, and makes testing much more precise, to be sure. But the unanswered question really is, do all patients with attenuated forms of MPS have elevated gags in the urine and dried blood spots at birth? We think the answer is probably yes, but the evidence thus far is really insufficient to conclusively answer the question, since newborn dried blood spots are very hard to come by on attenuated patients. And there have been efforts to gather as many as possible, and a few have been tested with elevated results, but there's still not really enough data to feel 100% certain. If they're not always elevated, then the use of GAGs for second-tier testing may be associated with a risk of missed cases. It has the benefit of many fewer babies having to come for testing, but there may be that small risk of missed cases. Now, what about MPS 2 newborn screening? This is currently ongoing in Taiwan, and as I mentioned, this is in the US only going on in all babies in Illinois and Missouri. In my state of Illinois, at the last tabulation, we had screened over 489,000 infants and found 6 babies affected with MPS 2. We also see pseudodeficiency for the MPS 2 enzyme. It's not as common as an MPS 1, but it is more common than true iduronate 2-sulfatase deficiency, than true MPS 2. So it's something that we have to sort out again with urine GAGs and molecular testing. But nonetheless, screening works for MPS 2, and I feel so gratified every time I think about these 6 babies who now have had the opportunity to be started on therapy very early in life, because I know their outcome is going to be good. It's going to be so much better than when we wait for clinical manifestations to appear. What about the other disorders? Well, we do have testing ongoing for MPS 3B, 4A, and 6 in Taiwan. So some data are being gathered there. And there is a Screen Plus program. It's called the Screen Plus program in New York under the direction of Dr. Melissa Wasserstein, who is doing expanded newborn screening for a large group of disorders not on the RUS in selected hospitals in New York with parental informed consent. And it currently includes MPS 3B, 4A, 6, and 7. So once data are generated through that program, we may have some pilot data that could support newborn screening for each of these disorders. There is also some screening for MPS 6 ongoing in Brazil. So what can families do to promote newborn screening? As I mentioned earlier, if your state is not yet in the process of implementing screening for MPS 1, find out why not and advocate for adding this to your state panel. It's been on the RUSP now for 5 years. You can also find out how new disorders are added to the panel in your state. Is it done through recommendations of a newborn screening advisory committee, or is it done through legislation? If your disorder is being discussed at a newborn screening advisory committee meeting in your state, attend the meeting and speak up. And if legislation is needed to add a disorder, or even if it's not needed, if it could potentially get action more quickly, you can contact your own legislator, discuss the issue, schedule a meeting, and advocate for legislation mandating the inclusion of your condition. Certainly if it's MPS 2, there's ample support for that now and data from other states. And with time, we will have data for the other conditions as well. So to summarize, newborn screening for MPS 1 is becoming widespread, and it allows treatment to be initiated at a very early age, improving the outcome and avoiding the diagnostic odyssey that is so common with MPS disorders. Newborn screening for MPS 2 is beginning and hopefully will soon become more widespread. Screening could be done for all the other MPS disorders. The technology is there, and families have been the main impetus for the initiation of screening and can be powerful advocates for moving this forward. With that, I'll conclude. Thank you so much, and we'll look forward to answering any of your questions.
Thank you, Dr. Burton, for that fascinating presentation. There's so much to know about this topic, And one thing I wanted to point out is that there are actually, just so everyone knows the relevance, there are families that are attending this conference today because they were picked up through newborn screening, and their children were treated at 3 months out from birth instead of at 2 years out from birth. And those children and those families will be forever changed because of the ability of having newborn screening. So this is a very real thing that really touches people's lives. Also, I want to remind everyone to add your questions to the chat for Dr. Burton, and we will take those at the Q&A session. So our next presenter is Matthew Ellinwood. Dr. Ellinwood began a residency in veterinary medical genetics and postdoctoral fellowship in comparative genetics with the esteemed Dr. Mark Haskins, we all know and love, at the University of Pennsylvania, where he began working on lysosomal storage disease models with a concentration on the neuropathic MPS disorders. He completed a postdoctoral fellowship in gene therapy in France and worked as an associate professor for many years at Iowa State University, where he began a long and fruitful collaboration with a number of colleagues in the field, including, again, the esteemed Dr. Patty Dixon, with whom he collaborated on MPS I and MPS IIIB research. He recently retired from teaching and his chairmanship in the Society's Scientific Advisory Board and joined the staff of the National MPS Society as our inaugural Chief Scientific Officer. We are so thrilled to have him on board on the Society staff, and we welcome him today, who will be speaking on newborn screening and why science matters. Matthew.
Thank you very much, Steve. And thank you, Dr. Burton, for a wonderful overview of newborn screening. I'll be capitalizing on quite a bit of the information that was presented both by Dr. Munzer and Dr. Burton. And I'll be going into a little bit more detail on the science and biology behind the tests that are being conducted as part of newborn screening. And my desire is not to turn you into experts of biochemistry or enzymology, but it's to try and give you information so that when you hear this alphabet soup of things like GAGs and IDUA, et cetera, that you have some information behind it so that it is not just meaningless strings of letters and words, but you have some sense of what it means. Also, as you go forward and advocate for newborn screening, you have a great deal of authenticity just because of your story, but having the authority of knowing about the tests and how the tests are done and how they're implemented can give you also a great deal of additional authority and authenticity when you go to advocate for these diseases. I also need to try and make an effort to make this presentation accessible for those who may be newly diagnosed, as well as for those who have been at more than one family conference. So I hope that I am providing engaging information for both of those diverse groups. So as we've heard from Dr. Burton, there is a RUSP nomination process, and that actually has a form. And the two main components of the form are divided. The first part really discusses the condition, information on the disease and the treatment. So what are the specifics of MPS II? What are the genetics, the biochemistry, the incidence in the population? What is the natural history of the disease? And then there is important that there also be an approved approach to therapy or an approved drug for therapy. And in this case, for MPS II, we definitely have an approved enzyme replacement therapy. How does that work? The urgency of the disease, that it be treated as early as possible in children. And for those of you in the family of MPS and ML disorders, you all know that it's a very urgent concern. The efficacy, the availability of the treatment, and any possible harms that might be associated with treatment. And Dr. Burton's done a wonderful job overviewing that, as has Dr. Munzer as well. What I'll spend most of my talk is the evidence-based information, the test that is being used, its validation, as well as how available a confirmatory testing approach is. And then we must have a prospective population-based pilot study of the disease where we've identified a patient, they've been confirmed, and they've gone on to be treated. And as Dr. Burton mentioned, that needs to come from data in the United States. So when we're talking about the validation of the laboratory test for MPS II, as Dr. Burton mentioned, this is going to be enzyme that is active in the newborn dried blood spot. And the enzyme we're talking about is iduronate 2-sulfatase. And this can be abbreviated as I2S, IDS, ID2S. You'll see all of those in the literature. Now, enzymes catabolize or digest a substrate, and in this case, it's something called glycosaminoglycans. You'll hear us call those GAGs. And in the case of MPS II, the GAGs that are digested are heparan and dermatan sulfates. There are other types of GAG, but they are not primary storage products. They're not the storage products or the substrates that are digested by this enzyme. So background, what are GAGs? These are long patterned chains of sugars. They're usually sulfated. There is one form that is not, and they're repeating disaccharide units. So here's a cartoon of a GAG, and these things like IDO, AB1, those are sugars, as are these GalNAcs and GLICNAcs. These are all sugars, and they're arranged in pairs, and they're repeating disaccharide units attached originally to a protein in the body, and they come to the lysosome for digestion. So again, these are disaccharide units, and they repeat. And these disaccharide units have either a uronic acid in green. So you've heard about iduronate or glucuronate. Those are the iduronic components. In MPS IV that stores keratan sulfate instead of an uronic acid, it's a galactose. The second sugar will be a nitrogen-containing sugar. And these patterned disaccharides repeat and then are attached to a protein. That's basically the biology of GAGs. The diverse type of GAGs that we see, heparan sulfate versus dermatan versus keratan, hyaluronan and chondroitin, these all depend on which sugars are present in the disaccharide units and the number and location of sulfurs, which you can see right here. So these are highly sulfated chains of sugars as well. Shown above here, this is a cartoon for dermatan sulfate. So, GAG degradation of enzymes involved in MPS, an overview here. This always proceeds from what we call the non-reducing end of the GAG. So this would be the GAG portion that is on the left. The portion on the right is the original portion that attaches to the protein. So all enzymes that are involved in the MPSs digest from the non-reducing end or the free end. So here we see iduronate sulfatase or iduronate 2-sulfatase, and it recognizes a specific chemical signature or a moiety that is exposed at the non-reducing end. In this case, it is a sulfur. So the enzyme here is depicted by those letters, and it cleaves off that sulfur. So the reason we have the name iduronate 2-sulfatase is that 2 is the second carbon in that sugar ring to which the sulfur is attached. Hence the name of the enzyme, iduronate 2-sulfatase. We're getting the sulfur taken off, or the sulfate group taken off of that 2 carbon. So in MPS II, this enzyme is not working, and it can't digest at the non-reducing end, and hence all of the other enzymes that are involved in GAG degradation, like iduronidase or aryl sulfatase B, they can't do their job because there's no free non-reducing end visible, so they don't see the moiety of the GAG that needs to be seen by that specific enzyme. As a result, GAGs accumulate. So How does a deficiency in I2S lead to storage of both dermatan and heparan sulfate? Here we just see dermatan. Well, that's pretty easy to answer. Because these sugars have similar patterns, some of them share specific chemical moieties at the non-reducing end. And in dermatan sulfate and in heparan sulfate, they both have sulfates that are attached to the 2-carbon of iduronate residues. So that's why we see both GAGs accumulate in MPS II. So the testing method will go after and look at this enzyme. And we have two main approaches, and those are both represented in the recent approach to testings that were validated in Illinois and in Missouri, and where we see newborn screening happening there. One of them is called tandem mass spectroscopy. Sometimes you'll see it as MS/MS. This was a technique that was developed at the University of Washington for the MPS disorders. And there are commercially available artificial substrates that are used for this test. And it measures essentially the mass spectrum, the amount of the size of a molecule from enzyme digested substrates. So it's incredibly detailed biochemistry, but effectively you're identifying a substrate that's cleaved from an enzyme by its mass, its atomic mass. The other approach is a fluorescence-based assay. And this has been developed as a commercial platform. In the laboratory, I used to do these assays as fluorescence-based assays, but there is a commercial approach developed by Babies in North Carolina. And it supports what we call digital microfluidics. We'll go into that. It's an amazing and fascinating approach. They also supply reagents for a traditional approach to MPS II, similar to the kinds of assays that I might have done in the lab when I was a postdoc and then a faculty. So the principle of newborn screening, it's going to be using a dried blood spot. The test needs to be quick, efficient, accurate, robust, and states are going to have to pay for this, so it needs to be economical. And it must follow a second-tier confirmation test that also needs to be widely available. The different platforms that I mentioned may be easier for different states to implement. The test must also maximize both sensitivity and accuracy. That means minimizing false positives as well as false negatives. So tandem mass spectroscopy, or tandem mass spectrometry, this uses an artificial substrate. So this is an iduronate sugar residue attached to an artificial compound. And when it is subjected to enzyme from the dried blood spot, it will cleave the substrate, and it gives you a special particular signature on a chemical signature on mass spectrometry. And we'll go over that. It is more sensitive than fluorescence-based assays. It can be what we call multiplexed. So in the same sample, you may be able to include substrates for multiple MPS disorders. And on the same batch or run, you may get data on all of these because the substrates will have different atomic mass and they'll show up differently. So that's one of the things that makes it very attractive. The knowledge and expense and room to house these sorts of approaches is not inconsiderable. So that may be a limitation to some states implementing it. This is the basis of the testing in Illinois, a big state, and they can afford that, and they have that approach used for other things. So give you an example of what these look like. Tandem mass spectrometry used on a 3 millimeter punch from a dried blood spot. So here is the substrate. You see circled in yellow, the sulfate attached to that sugar ring. And then this large, strange organic chemical compound is the signature molecule they'll use to identify on mass spectrometry. The enzyme from the dried blood spot circled in blue will cleave this, and you will see the sulfate released. It's gone. And then this substrate will have a particular pattern. What you get as a result will be traces of atomic mass that are recorded by this mass spectrometer. So an example here in lavender is the internal standard that's included for the iduronate 2 sulfatase enzyme assay. And then in the green trace, you'll have the product. So you have the substrate that's released and the original substrate before it was cleaved. And then you have the substrate that's uncleaved. So it's the changes in these peaks that tells you how much enzyme is there, in which case a patient is normal, or how much is not there, in which case the patient may be at risk for MPS II. That's the basic test approach seen in tandem mass spectrometry. Fluorescence-based enzymology is the approach that I used when I was a researcher, and it basically allows a free fluorescent compound, or fluorophore, to get attached to the iduronate 2 compound, and then you can measure fluorescence. So what we have here is the iduronate 2 sulfate in green, and it's attached to this molecule above it, which is a fluorophore. When they're bound together, it does not fluoresce. The fluorophore, unfluorescing because it's bound, is something called 4-methylumboliferone. If you've been at many of these talks, you may have heard people talk about 4-MU. The enzyme comes along and cleaves that, but you notice that we don't have any cleavage of the substrate. That's still uncleaved. For the ID2S enzyme assay, it does need iduronidase added, so that will liberate the fluorophore from that compound. And then the liberated fluorophore will fluoresce, and you can measure it as free 4-MU. So in the assays, you'll see nanomoles or micromoles of 4-MU or substrate. That's what they're measuring. So there are 2 platforms currently being used that employ a fluorescent-based approach. Digital microfluidics, which is being used currently for MPS I, and a conventional, what we call 96-well assay platform. This is the way I used to measure enzymes. These are both developed by a company here in the North Carolina area called Babies, a spinoff from Duke University. And the digital microfluidics platform is a miracle of engineering. It's currently used for MPS I, and it relies on very small volumes from a dried blood spot sample. The sample is incubated in this disposable cartridge here in orange. These cartridges can load up to 44 samples each, and they can test up to 5 tests per cartridge. So for a small state that doesn't have a high birth rate, these can be very attractive approaches. The sample droplet is actually moved specifically through this cartridge with electrodes that are embedded in this disposable cartridge and along channels of the plastic that have been coated to make sure that the droplet follows its appropriate path. It is rapid, it is self-contained, and it is an FDA-approved machine for testing of lysosomal storage diseases on newborn screens. So a real marvel of engineering. Currently in Missouri for MPS II, they're using a 96-well platform. It's shown in black here because you can't have fluorescence from one well spilling into another. And it is the basis for all of the data that's been generated on MPS II in Missouri. And we do have a digital microfluidics platform that is being developed by Babies for MPS II. So hopefully that will be available shortly. So test validation, this is required for any test used, and it employs dried blood spot control samples that are supplied by the Centers for Disease Control. And these are ostensibly a normal population of cells, and then using known affected dried blood spots. We need to make sure that the assay has something called linearity. So over a broad range of concentration, we get reliable reporting of enzyme activity. There needs to be good sensitivity and precision, and it must be clinically validated. So this was a paper out of Missouri on the clinical validation. I'll go through more detail on the results from Missouri because I think they're more accessible. But we also have data, and this used the fluorescence-based assay, but we also have data from Illinois on the development of the assay in Illinois, and we'll go over some of the results from that as well. So assay validation, as I said, we need to show linearity, which is that over a broad concentration, we get a reliable output of signal. See you here, micromoles of activity of 4MU or of 4MU released per liter per hour. And these are the concentrations. If we have a perfect correlation, this value here, R squared equals 1, and we got pretty darn close to that. So this is a good assay. We also need a widely available second-tier test. So the basis of confirmatory tests is a confirmed deficiency of the enzyme, elevated heparan and dermatan sulfate, which can be assayed in the dried blood spot or in urine. We also need assay of a second sulfatase, because there's a disease out there called multiple sulfatase deficiency, which can lead to low IDS activity. And in that case, you need to test another sulfatase. If it's normal, that supports a diagnosis of MPS II. And most places will also incorporate genotyping for a mutation. Samples that these can be run on include dried blood spots, urine, and plasma or whole blood for a second sulfatase. And these are available widely. There are over nearly 90 laboratories that are licensed and able to do these. Mayo, Greenwood Genetics Center, Perkin-Elmer, University of Chicago, or Illinois, Chicago, and Duke are just a few. The assay needs to be clinically validated. So we need to have a test of a population, a clinical population, with known affected dried blood spots. And this was done in Missouri. So they had a presumptive population here of normals, and then they added to that some known affected dried blood spots. So those are the normal populations, presumptively normal, and then they had here some known affected dried blood spots. So you can see a really wide gap between the tested affecteds and the normals. So looks like good assay clinical validation. We need to assess the risk of false negatives. And what they did was they looked at the standard deviation of the affected samples, and that was very low. And they compared how many standard deviations, which is a measure of variability, separated the highest affected from the lowest normal. And it was 25 standard deviations. So we got really good discrimination between the lowest normal and the highest affected from this sample. But this was not necessarily a prospective population test. So this is the graph that shows the population-based test at Missouri. And it was done from 2018 to 2020. And they did approximately 150 newborn dried blood spots. So their retest was 25 micromoles per liter per hour. Micromole is a measure of how much 4MU they could evaluate. And that prompted a retest of the sample. And then they would look at if it was below 20, that is what they called a potential at-risk allele. So anything below that line there. So these are what they have for results from Missouri. They tested nearly 150,000 newborn blood spots. 100 of them roughly required a retest. And after that retest, they identified 44 at-risk alleles. 12 of those were considered low risk based on results of previous screens or other abnormalities in their dried blood spot. Second-tier testing was done on 29 samples. And the results of those 29 identified 2 affected children. So 1 per 70,000 roughly. There were 27 false positives. 9 of those were normal. 3 had pseudo deficiencies, 10 had variants of unknown significance. That's a genetic change, but we can't predict what it's associated with. They did not have elevated GAGs, however, and there were 5 at the time of publication who had testing that was still pending. So the results from Illinois, I'm taking information off of the most recent publication from Dr. Burton. She presented some more recent data. So the numbers from the earlier iteration of this test was 340,000 roughly. They found 31 at-risk alleles, and they found 3 affecteds. I missed a digit in there. That's roughly 1 in every 100,000. That number has changed slightly from what Dr. Burton presented today because she has more information. There were 28 false positives, 3 normals, 25 pseudodeficiencies. So we do see pseudodeficiencies in MPS II. They're not as common as they are for MPS I, but they do exist. But still, excellent discrimination on this, and these numbers are part of what we need to submit to the RUSP. So how does that process work? The nomination was drafted, and we assembled a panel of clinical, medical, and genetic experts with biochemical expertise in the diagnosis of these diseases and in the use of newborn screening testing platforms. And you'll recognize some of the names on that panel. Dr. Burton, Dr. Munzer, Michael Gelb, and Ron Scott at Washington, who helped develop some of these assays, as well as clinicians from other parts of the country who were expert in MPS II. So the initial submission was in spring. We worked through the office to develop, to address some of their editing concerns. And in December, a final revision was submitted to the Advisory Committee on Heritable Disorders in Newborns and Children. Again, that was that committee that Dr. Burton mentioned that makes recommendations to the Secretary of Health and Human Services. Their review was delivered on May 13th when they recommended that the nomination be forwarded to evidence-based review. So that's where we are now. We have 9 months to hear back from them. And really, MPS II has fulfilled all the requirements. There is a test, there's urgency, there is both specificity and accuracy to the test. We can back it up with confirmatory diagnostics. So hopefully we will see this is our second MPS disorder that has been approved for newborn screening. I would like to thank the nominators, members of the expert panel, and that concludes my talk. I hope it hasn't been overly sciencey and that everybody has been able to bring something new away from the information that we've presented today. Thank you so very much.
Thank you, Dr. Ellinwood. That's a very tremendous amount of detail and good information for all of us to digest. And just put a reminder in here for everyone, all of these sessions are being taped, and you can view those for the next 30 days in case you want to go back and study some of that a little bit in more detail. Again, a reminder to ask your questions by submitting a question on the screen, and that'll go into the queue so that when it's time for the Q&A, we'll have those all ready to run with. Our next speaker and our last speaker before the Q&A is Dylan Simon. Mr. Simon serves as the Newborn Screening and Diagnostics Policy Manager for an organization that is very dear to all of us, the Every Life Foundation for Rare Diseases, which was founded by Dr. Emil Kakis. He advocates for federal and state newborn screening legislation, creates advocacy tools for patient advocates, and maintains the Foundation's Newborn Screening Action Center. Previously, he interned for the Senate Help Committee and advocated for Research America and Veterans Against Alzheimer's. Dylan received his master's degree in molecular microbiology and immunology from the University of Maryland. We welcome Mr. Simon, who will be speaking on working with state advocacy to pass newborn screening legislation.
Hello and good afternoon. My name is Dylan Simon. I'm the Newborn Screening and Diagnostics Policy Manager here at the EveryLife Foundation for Rare Diseases. I want to thank you guys for having me here today. I'm really excited to talk to you about the importance of working with state advocates to pass state newborn screening legislation. And so today I'll start off with a little bit about the importance of state level advocacy, as well as in discussing a little bit about the work that we do as a foundation at the state level, and close it with the importance of a coalition in passing that state newborn screening legislation. Again, so I want to start with the impact of grassroots advocacy. And the reason I want to start there is because I really want to highlight the fact that you guys do have the power to affect change. Whether as an educator or trainer, you are a great resource to both federal and state legislators. And that really at the end of the day is because you are your greatest advocate. You know your story the best. As somebody who works based in DC, when speaking both with federal and state-based legislators and staff, I can give them all the facts and figures and I can relay your story to them. But you know your story the best, and they want to hear from their constituents more than they want to hear from me, because every effort to advocate matters, and hearing your story is what's really important to them and really what drives home the importance of any legislation, whether it's newborn screening or any other rare disease related legislation. Hearing that story from the constituents, from the advocates, is what's really important to legislators and their staff. And I think it's also important to know that there is a role for every advocate. Not everybody's comfortable in every setting. And so it's really finding that way to advocate for yourself that you're most comfortable with. And so we'll go through at the end a couple of different ways that you can do that. But I think it's important to note that if you want to get involved and you want to affect change, you want to speak up and tell your story, there's a wide variety of ways that you can do it. But when it comes down to it, anybody who wants to advocate can. It's just about finding the way that works best for you. And going back to what we talked about before, why relationships matter. And so you can see at the very top, in terms of what has positive influence or subtle influence, the number one answer is in-person visits from constituents. Now, obviously, this was taken prior to the pandemic, and so in-person visits haven't been really an option the past year and a half. That hopefully will be changing in the coming months and years. But you can see the second option is still contact from constituent representatives. And so whether or not it's an in-person visit, just hearing from their own constituents is really important for representatives. And as you start to go down the list, you see what really matters is the relationships and these individualized contacts. And so whether it's in-person visit, contact, a letter, an email, an individualized letter, an individualized email, what they want to do is they want to hear from the individual, the advocate themselves. As you start to get into more broader themes and broader ways of contact, that's where you start to lose some of that influence. But this really highlights why relationships are so important when advocating at the state level. And again, the importance of state legislative advocacy. The state in which you live can dictate the type and quality of care that you have access to. Many healthcare regulation bills that impact rare disease community are made at the state level. Newborn screening will go into a lot of detail into that about the state impact of newborn screening programs. But that's across the board throughout healthcare in terms of you can have great impact on the community through state level advocacy. In addition, it increases awareness for resources for rare disease diagnostics, treatments, and access to care and coordination of care, as well as creates state community action, legislative action, has the potential to improve healthcare delivery for the full community. And so it's always important to remember that state and federal advocacy both have their role because state and federal legislators both have their role. And each can have a profound impact on the rare disease community. So if you have always been active in one, there's always opportunity to try the other. But at the same time, if you can only be active in one, or if you're only able to be active in state legislative advocacy, that's perfect. I mean, that's fantastic. You can still advocate for the issues that are important to you because so much of the healthcare for the rare disease community is decided at the state level. And so I just want to go back a little bit and talk a little more in detail about newborn screening. And so just a little bit of facts about rare diseases. There are 400 million rare disease patients worldwide. About 50% of them are children. And rare diseases are responsible for about 35% of deaths within the first year of life. And so this highlights not only how large the rare disease community is, but how impactful newborn screening can be. The ability to identify these conditions early in life and increase the access to treatment early in life where they can really make a difference is really at the core of newborn screening. And so newborn screening reaches each of the 4 million newborns in the US every year, and approximately 1 in 178 has a condition that can be detected through newborn screening, which tests for a variety of genetic, metabolic, and hormonal conditions that are not otherwise apparent at birth. And I said this before, and I'll say it now, and I'm sure I'll say it again, but this last box really highlights the fact that newborn screening facilitates the timely delivery of life-saving treatments and other therapies that improve the quality of life. So we'll talk a little more in depth about this idea, but at the core of newborn screening is the delivery of these treatments when a newborn is asymptomatic and you can stop the degenerative nature of those conditions. In addition, newborn screening helps to trim down on that diagnostic odyssey. And so it takes on average 16.5 years in the United States to obtain a diagnosis for a rare disease since their first symptom. And 40% of patients receive a misdiagnosis more than once. And on average, patients see 17 physicians before receiving the proper diagnosis. And so newborn screening provides an opportunity to eliminate this diagnostic odyssey that so many individuals in the rare disease community have to go through just to get a diagnosis, just to understand what is happening to them and the condition that they have. In addition, 70% of physicians believe it would be helpful to have additional training in rare diseases. And so the doctors want the help. They want the information about these conditions. And newborn screening provides opportunity through its educational process within the program. And we'll talk a little bit more about that later. But again, going back to newborn screening, it provides an opportunity to get that early diagnosis and cut down on these 17 physicians and that 16.5 years to really get the diagnosis and start treatment as early as possible. And so I want to talk a little bit about how a condition can get on a newborn screening panel. And so what we're going to talk about a little bit right now is the federal newborn screening program. And then we'll get a little bit into the state program. So as you can see on the bottom left, it might be the small circle, but the evidence development is the longest part of this process. And so you have to develop a screening method to properly identify a condition. You have to ensure that there's a therapeutic available. You have to ensure that there's an infrastructure in place to conduct the follow-up visits, as well as care standards for those conditions. So this process can take 5 to 10 years, if not longer, to really develop each individual part of that. And during that time, often leading the charges are the patient advocacy organizations. And so I don't want to dive too much into that part because that is a completely different conversation, one of which I would always be happy to have. But in terms of this conversation, it's just important to note that advocates are involved in that process as well. They can step in and have their voice be heard, help to share their data, help to work with patient advocate organizations to gain access to clinical trials or pilot studies to help develop that package. But during that process, the patient advocacy organization is bringing all the information together so that they can submit for a RUSP nomination package. So RUSP stands for the Recommend Uniform Screening Panel. It is the federal recommended newborn screening panel that is overseen by the Secretary's Advisory Committee on Heritable Disorders in Newborns and Children. So the Federal Advisory Committee not only sees the RUSP, but oversees the overall federal newborn screening program. And so I think it's important to note that the federal program very much supports the state program. And so the Secretary Advisory Committee is really at the core of that. It is reviewing what are the issues within newborn screening that has a federal solution, not just a state solution. And so looking at workforce issues or looking at, for instance, coming out of the pandemic, what were some lessons learned across the country on how to deal with a public health emergency with newborn screening and how to take lessons learned for the next public health emergency. There may not be another pandemic, but public health emergencies will always come about typically through natural disasters. So if there's, let's say, a hurricane in Florida and it creates a public health emergency, how can we take lessons learned from the pandemic to implement to ensure that newborn screening is not interrupted. But going back to the RUSP, once the patient advocacy organization submits the RUSP nomination package, it is then goes under review. The first review is whether or not it should go into the evidence review committee. And you can see here, if the package is not accepted, you can continue to build evidence. And you can see multiple times throughout this process, there are times in which the package may not be approved. But at each step of that process, it is not a forever no. There are multiple conditions that are currently on the RUSP that submitted multiple packages to the committee. And the committee is very open with patient advocacy organization, and they want to have an open dialogue, on how to build the evidence needed for RUSP approval. And so, and as happened on a few occasions where they went back, they conducted additional studies, collected additional data, resubmitted the package, and it was in fact added to the RUSP. So once it goes into the evidence review committee, they then provide their report, which is then reviewed by the advisory committee, which is separate from the evidence review committee. Once they then review, the advisory committee then makes their recommendation, which then goes to the Secretary of Health and Human Services. From there, the HHS Secretary will then approve the condition to the RUSP, and that's when state-by-state implementation begins. So as I said, there is the federal and state program, and I think a key part of that is the fact that the federal RUSP is just that, it is a recommended panel. The final decision on what each individual state screens for lies with each individual state. And so once a condition is added to the RUSP, at the moment, the next phase is just beginning for the patient advocacy organizations. They are now have to go and begin that state-by-state implementation process of going to each individual state to then ask them to add their condition to their state panel. And I think it's always important to kind of look at the key considerations for when adding a condition to that RUSP. Because I think it helps to kind of frame what the advisory committee is looking at and what exactly each package needs to contain. So first, the screening needs to identify all babies who may need treatment. So very simply, can you identify the newborn who has the condition within the general population? In addition, can you try to limit the false positive, false negative rates? And so there is a rate that the evidence review committee will find acceptable. But essentially, at the end of the day, it's can you identify all babies who may need treatment without misidentifying those who don't? In addition, they're looking at is there a significant risk of illness or disability or death if babies are not treated promptly? And this goes hand in hand with point 3, as well as 4, with effective treatment is available, as well as treatment is more beneficial in the newborn period rather than later. So this goes back to this idea of asymptomatic treatment. So at the core of newborn screening is this idea that these conditions will worsen over time, and the damage that is done is not reversible. And so identifying them as early as possible, beginning that treatment to eliminate the condition and the repercussions from the condition from starting to set in is a really key part. And so can you identify the baby at an early time? And does that condition carry a risk of serious illness or death? And do you have the treatment available to ensure that that does not happen? And so that really is kind of that key part of newborn screening, is the asymptomatic life-saving treatment that can occur from the early diagnosis. In addition, spoke previously to that, resources and access to treatment and counseling are widely available. We want to make sure that if you are identifying these newborns through the diagnostic testing, that they can be brought back in for confirmation testing, and that there are resources available to support that family through that diagnosis and through the early parts of treatment. In addition, the benefits to the babies and to society outweighs the risk and burdens of screening and treatment. So this goes back to that false positive, false negative rate, is are you able to properly identify those who need treatment without falsely identifying those who do not, or missing those who do? And so I think it's always worthwhile just to really look at what the RUSP process is really looking for when considering whether or not it should be added to the federal recommended panel. So that brings us to the state newborn screening. And so as I said, a lot of newborn screening is done at the state level. And so you can see here, the the number of conditions screened, that's decided at the state level. Whether or not there's an advisory council that oversees that state program, that varies state to state, but many states do have it. In addition, how is it funded? The screening fees, that where the actual lab does the testing is all decided at the state level. And they also decide when to add a condition. So currently it takes on average 5 to 6 years to add a condition following its addition to the RUSP. And so we'll talk a little more about ways that we want to improve that number in the coming slides. But I think it's always worthwhile to really identify why that occurs. So first and foremost, adding a condition often occurs through legislation, and that can take some time. But when speaking to the labs, I think it's always important to know why they're saying that it takes that time. And often it's talking about making sure that they can properly implement the new testing. They want to ensure that they have the funding available to either hire the new staff or to buy the new machines or to do the property quality assurance associated with newborn screening programs. They also want to ensure they have the follow-up service in place to ensure the fact that they can, not only can they properly identify through the diagnostic testing, but that they can get back in contact with the family and bring the family back in for confirmation testing. So all of that does take time, and we do understand that to ask a state to immediately add a condition once it's added to the RUSP is asking a lot of the state, because it is not, they do not have the facilities, they do not necessarily have the personnel just waiting there ready to go. And so it does take some time, and it's always worth noting why they say they can't add it immediately, even though we hope they would add it a little sooner, which we'll talk about momentarily. But again, at the end of the day, there's so much to advocate for at the state level surrounding newborn screening. And it really comes back to not only the conditions added, but also the education part of it. And so newborn screening is often viewed as just a test, but is really a full system. So we've talked a lot about the follow-up side of it, but there's also an education side of it. So ensuring the fact that there is educational materials available for families when they've received the diagnosis, ensuring the fact that there's education materials for the healthcare professionals. Again, going back to that one of the earlier slides, physicians just want more information on these rare diseases. And so part of the newborn screening program, especially at the state level, is to provide that education, working with healthcare providers and healthcare professionals to ensure that they are educated on these rare diseases, so that when they are giving the diagnosis to those families, they can provide the proper information about that condition. And so now we get to the state legislation that we work on. So we call it RUSP alignment legislation. And so some states screen for as few as 30 conditions, other states screen for more than 60 conditions. And programs vary widely by state, leading to a wide variety of health outcomes. And so we pursue RUSP alignment legislation. And this year we pursued it and we try to lead the efforts in 3 states. So what exactly is RUSP alignment legislation? So first and foremost, it requires the state to screen for any disorder recommended by the federal RUSP. It does not preclude the state from screening for conditions that are not on the RUSP, but too often the RUSP in today's world is the goal. And with RUSP alignment, we want the RUSP to be the minimum. We believe that if a patient advocacy organization can pull together a package and go through the advisory committee's science-based, evidence-driven approach for addition to the RUSP, that the state should then be able to screen for that condition. And so the first part of the legislation is that requirement that if it is on the RUSP, then the state must screen for it. In addition, it implements a timeline for when the state needs to begin screening for new RUSP disorders. But as I said, currently it takes on average 5 to 6 years to add a condition to a state panel once it's been added to the RUSP. Now, while I said before, we appreciate that you cannot add it within 6 months, we believe that 5 to 6 years is too long. And so typically we advocate for about a 2.5-year process. And we believe, after speaking with multiple state programs, that is a timeline that is reasonable for them to ensure that they can properly administer the test while also doing it as quickly as possible. In addition, we want to ensure that if we're going to require them to screen for a condition and we're going to require them to do it in a certain amount of time, that there is a funding source available to them, that there is funding to buy that new machine, to hire the new staff if needed. And so it's working with each individual state to really identify what is the best way to do this. And so I think it's important to note that we go in, when we go into these states, we go in with these themes, these ideas. We don't go in with a single piece of legislation and hand them like, you need to pass this this bill as written. We want to work with each individual state because each individual state is different, and they have a different state constitution. They have different norms. They have different politics. They have different legislators, and you have to be able to be adaptable and work with them when possible, and use these elements as your shining goal, and continue to make sure that you're reaching them, but working with the state to figure out how best to do that. So again, in this past year, we have been leading the efforts in 3 states. And we're really excited that in Georgia, we were actually able to sign the bill into law on May 4th, which we are very excited about. And we're currently pursuing legislation in Ohio and North Carolina as well. In Ohio, it was included in the budget language that passed the House. It is currently included in the Senate budget language. I will say that I encourage you all to ask me in the Q&A to give you an update on both Ohio as well as North Carolina, but because I do know for Ohio specifically, there should be an update between when I'm recording this video and when I am there for the Q&A. So I'm excited to hopefully give you a wonderful update at the end of this session. And in North Carolina, we were able to pass the legislation at the State House of Representatives almost unanimously, and we're currently waiting scheduling in the state Senate. And again, I encourage you to ask me for any updates because hopefully I will have one for you when we do the live Q&A for this. So now I want to talk about kind of the state, how to advocate for policy. And so it really gets down to creating a relationship with your state and federal legislators. Share your story with a policy ask. So it goes back to this idea of you are your best advocate. How can you share your story, but also couple it with newborn screening? So sharing your story, and because of this, this is why this legislation is needed. Here's how it will make real impact in the lives of your constituents. In addition, ask questions. At the end of the day, your legislators are individuals who are curious and want to do good and want to help. And so ask questions, talk to them, see how you can be helpful to them. Because at the end of the day, you want to be a resource to them. You want to bring your story, but you also want to be able to be a resource for them to come back to continue to ask questions and continue to develop support for your ask. In addition, support others with their policy goals when they align. I think that's important to note because we'll talk a lot about coalitions coming up and the importance behind them. And so when possible, work with each other, work with your rare disease advocates as a whole, because the rare disease community, when spoken with one voice, is a very loud voice. And so working across advocacy communities is a really great way to advocate for your policy. And going back to that, advocate for your own network. You have one. Bring in your family and friends. Work with the other rare disease communities. Work with your friends and family. Activate your own network and bring them to the advocacy world. And again, there are different ways to engage. And I'm going to go from the bottom first and make it personal. I've said this multiple times, and I'm going to keep saying it. But sharing your story is such a key part of engaging with the legislators. And going back to what I originally said in one of the first slides is find the tactic that works best for you. Is it an in-person meeting? Is it a briefing? Is it a letter? Is it a phone call? Is it social media? Is it writing a letter to the editor? There's so many variety of ways to get involved. If you want to get involved, there's a way for you to do it. So it's really just identifying what works best for you and committing to that and getting involved and engaging the legislators. And so specific to our own effort, it's always important to identify the key stakeholders within the community. So within newborn screening, you can see there are so many key stakeholders, but when building a coalition, there are really 3 main avenues. There's the child, the parents, and the patient advocacy group. And so you want to work with the local community to build support for legislation and contact legislators. And so you can do that in a variety of ways. And so the first is something called action alerts, but in general, contacting your state representatives and senators. And so this is an example of one that we're currently using in North Carolina. And it provides an option to create an automated way to contact your representative. And so, as I said, you want to individualize that when possible, but you also want to help other advocates where possible. And so providing information on the legislation, providing them with the sponsors of the legislation, and providing them some information on the impact of the legislation. But while providing an opportunity for them to individualize that communication. And so these action alerts provide an opportunity to contact your state reps. And so this is essentially like an email. And so you can always do individualized emails, and I highly encourage them when possible. But having these broad action alerts is a great way to work as a coalition to send them into various state senators and state representatives. In addition, community letters of support. This is very small, fine print. This is not meant for you guys to be able to read the actual letter. But the idea of the letter itself is to send it to the leaders on the Hill in the state legislature and highlight the support for the program. Legislators want to know that there is a support for the bills that they are advocating for. And so the key part of this slide is on the right side, which is you can see is a list of approximately 50 patient advocacy organizations who all support the bill. And this is really great to get that letter to the representative to highlight, thank you so much for your work. Thank you for advocating for this bill. Thank you for including the policies that will make an impact. And here are all the organizations that support your work. And again, speaking to legislators. So not everybody is always confident or want to participate this way, but again, there's multiple ways to do that. You can email, you can call. Hopefully soon, in person will also be an option. In addition, it is an option to request to speak at a hearing. We in North Carolina, we had an advocate speak, and she was fantastic. And we've had also in Ohio, where you can submit written testimony. That's another way that you can speak to legislators. But requesting to speak at a hearing is a great way to speak in front of a larger group of legislators and highlight the impact of the legislation. And again, social media as an advocacy tool. Use social media to your advantage. Make them aware of the problems that you face. Do not forget to thank them for their actions, especially at the state level. There's so much going on, and generally in a very short time period. And so be sure to thank them. And you can see here on the example, we tagged the lead representative on the bill who helped us get there. And so you always want to be advocating for your issue, and you always want to be thanking your champions for the issue. And again, I just want to thank everybody for your time today. I hope I was able to help walk through kind of the importance of working as a coalition at the state advocacy level. And I look forward to your questions today. Thank you guys so much.
Thank you, Mr. Simon, for that very good presentation. I want to especially send my thanks to you and the Every Life Foundation for the incredible work you're doing on behalf of the patient advocacy groups. There's no way we could be successful in this area without your intense effort and knowledge. So thank you for doing that. It's now time for our panel, and we have all three of our speakers here with us. If you haven't already, then please submit your questions in the Ask a Question feature, and we will get those and we will try to address those. We have 15 minutes scheduled to do that. So the first question I'd like to address is for you, Mr. Simon, and that is, based on your presentation, if we have a family that lives in one of these states that is not currently screening for MPS 1, even though it's on the RUSP, and they want to get involved, do you suggest that they just jump in with both feet, or would it be helpful to coordinate with either society or Every Life Foundation, or how would they go about doing that?
Thank you for the great question and for having me today. I think the best way to put it would be to jump in with both feet, but how that looks is going to vary on the state level. I think I talked a little bit about it, but the importance of having a coalition is always going to make everything a little easier. So I think touching base with the National MPS Society is a great place to start, just to kind of touch base and see what's being done, because you never know if there are previous efforts that are occurring that you can just join, as opposed to are you starting out on your own. And so I would definitely, whenever starting, I think it's always helpful to touch base with other organizations and see, okay, how can I start building this coalition of advocates together to get MPS 1 added to the state panel?
Steve,
okay.
if I
Sure.
Could I just interject a comment here? I think also that it would be helpful if there is a family in that situation to talk with your geneticist locally, because they will know how the process works in your state. In other words, is there an advisory committee? Does it require legislation? So they can probably give you some good information about at least where to start in terms of, who you could contact. But I think, someone in the Department of Health, I think it's always helpful if the people in the Department of Health know that consumers are watching, and they care about the issue and they're concerned, just calling and saying what's going on. We live here, we have, an MPS disorder. We see that all these other states are doing screening. We're not. Can you fill me in on why not? And in my experience as a professional, having been involved in some of these legislative efforts, I think in individuals in state government, legislators, they all listen so much more to the patients, to constituents. The fact that they know you're concerned, you're watching, carries a tremendous amount of weight, much more so than for having a professional to tell them that we need to be doing this. So, yeah, I just wanted to mention that.
Yes, thank you.
I 100% agree with that. And going back and kind of piggybacking off that point is speaking with legislators, they want to hear from their constituents. So I think always starting with your representative is always a great place to start. Mm-hmm. And that'll also help if you want to reach out to the Department of Health and newborn screening program, because the newborn screening program is busy and they tend to want to respond to people. But they're definitely more likely, fortunately or unfortunately, to respond to a legislator who's reaching out to them with questions. And so that's always a good first place. And when doing that step, I would definitely come at it from a fact finding. Like, I'm just curious. I want to know why this isn't happening. How can I help you? It's when approaching, you definitely kind of want to start with, okay, how can I be of help? And not necessarily accusatory, like, how can you not be screening for MPS 1 yet? Definitely start with why is this not happening? Let's work together to figure out the information. And then once you have all that, then you can start working to, okay, I get all this, but this needs to change, and here's how we're going to change it.
Okay, great. Great.
I have a question for Dr. Burton. If you look at that map that you displayed, many of the states that are represented may be in the rural West, not have large populations. Is there information that we can get that shows what states are part of consortia where you have multi-state testing programs? And if we knew that, we could connect families in those different states so they could lobby effectively for that group of states to begin testing for some of these diseases.
I'm not sure where that information is housed. I mean, I know personally of some. We have the New England Consortium, but we have a lot of screening going on up there. You're right, a lot of the ones that aren't are these more less populated, more rural Western states. Some states do utilize Oregon for their newborn screening. So I don't know exactly which one. I see Mr. Simon is nodding. He may know which ones, utilize Oregon. We certainly could find out from them. But I don't know the answer, I guess, is the bottom line.
I don't have the information off the top of my head, but I can actually let you guys know where to find it. If you go to our Action Center, our Newborn Screening Action Center, which is rarescreening.org, we have a state map where you can see which lab does the testing. And so that can help to identify, for Oregon and Washington, I know do screen on behalf of 4 other states. I will say, as important as that is, and recognizing those states, it still comes down to the individual state on what they screen. So let's say, for instance, I know Massachusetts does a screening for Vermont, but Vermont is the one who's deciding what Massachusetts is screening for the Vermont babies. And so while it's definitely helpful to know which of these hub states are what they're screening for, Iowa's a great example. They screen for a few different states, and they only currently screen for 31 conditions. And so that obviously is going to kind of hold back some of the other states. But for Massachusetts and Washington, for instance, where they screen for all 35, the states that they screen on behalf of, those states are still making the final decision on which conditions are being screened by Washington.
Okay, great. And for all these questions, I'll probably direct it to someone, and then just as you did, everyone else pipe in afterwards. So this next one, I think I'll start with Dr. Burton. If my state doesn't screen for MPS at birth, what tests do I need to do when a sibling is born? That's part A of the question. Part B of the question is, if I have an affected child and my state does screen, is it advisable to rely on that screening for a sibling, or should I do more than that?
Well, I think you probably can rely on the screening, but you're going to do more than that. And typically, families like to get the most rapid information that they can. So I recommend, whether your state screens or not, either way, work through your local physician, most likely your geneticist, and they will do the appropriate testing on a new sibling as soon as they're born. Usually that's going to involve the enzyme assay and likely the DNA testing as well. And I think the same would be true if your state does newborn screening. We would anticipate that you would get a positive newborn screen, but certainly you can proceed with the testing even before that occurs, and it can be expedited so you get the most rapid early diagnosis possible.
Okay, thank you. Another question, and this is for any of you: what is the status of newborn screening in Europe? Are there any countries leading the effort there?
I would say generally Europe is not as advanced as the U.S., that we screen for more conditions overall in the U.S. than in most places in Europe. And that certainly is true for lysosomal storage disorders. There are some patchy screening programs in various locations in Europe for lysosomal disorders, including MPS disorders, but we don't see the same kind of widespread newborn screening that we have here.
I would agree with that. I will say it does seem that they are trying to improve, specifically within the UK. There seems to be a more of an effort starting to arise. I will say on the international stage, outside of Europe, Taiwan tends to be a leader in newborn screening. But definitely, I would agree with Dr. Burton. Europe is kind of a patchwork at this point.
Yeah, Taiwan and also Japan. Japan has quite a bit going on with the MPS disorders, with newborn screening. Again, it's not countrywide, but they have quite a few programs in various regions of Japan.
Okay, thank you. Can one of you describe sort of the emotional support that's provided? So once a family has gone through the preliminary testing, then the confirmatory testing, what is sort of the next step in getting help for those families?
I can speak to the society's efforts in that. And we have a wonderful program called Pathways. So we have a dedicated social worker, and their position is to engage with these newly diagnosed families the minute we hear about them. And before COVID, they were actually traveling out to these families and spending a week with them. So the diagnostic journey and the way it's received by the families is quite different now with newborn screening versus a situation where the family knew something was wrong for some years before they got diagnosed. So that requires a different approach. And we have, again, our social working staff is really adept at working with those families who can't see a disease in a child. And they get this diagnosis at the newborn stage, and going out there as part of Pathways. And we stay with them in Pathways for an entire year. And then they graduate on to other aspects of support. But I think one of the really wonderful things about the society that's been initiated recently is this program to help support these newly diagnosed families and especially meet the challenge of a newborn screening diagnosis.
Great, thank you. Anything else to add from anyone?
No, not really. I think that is tremendously helpful. So we always encourage families as soon as we make a new diagnosis to contact the National MPS Society. I think, of course, at the local level, we try. We have social worker in our program, for example, who's also available to our patients and families, and genetic counselors. But it is just a very turbulent time. And I do think the newborn screening situation is very different. It's difficult for families to look at what looks like a perfectly normal, healthy baby and have somebody tell them that that child has a condition that they've never heard of. And, it's just all very overwhelming in many cases. So I think the program that the National MPS Society provides is just incredible and very beneficial.
Okay, thank you. Another question, and this probably is either Matthew or Dr. Burton. What does it mean when my paperwork says variant of unknown significance on the DNA results?
Scientists hate to say they don't know, so that's a fancy way of saying we don't know. I'll let Dr. Burton take this further on.
Okay.
I mean, I think it's really important that you know the context of how you get that result. I mean, what it basically means is that there's a change in the gene, but it's not one that's previously been described in a patient with the disease, nor is it one that's known to be just normal variation in the population. But we know with the MPS disorders and with many other genetic conditions that patients and families may have their own unique mutations. They may have mutations that have never been seen in another patient. So if you have a patient who looks like they have MPS II, for example, and they have elevated urine dermatan and heparan sulfate, and they have low I2S activity, and there's a variant in the gene that's described as a variant of unknown significance because it's not been previously reported in a patient with MPS II, you can pretty much assume that that is their MPS II mutation. On the other hand, if you see it in a newborn who has a positive newborn screen but normal urine gags, that's probably a pseudodeficiency allele. But because this is not a disease where everybody has the same change in the gene, we have to interpret these unusual gene changes in the context of the clinical setting. So, that is not an uncommon situation for that to occur. It's actually very common. And the interpretation of that really depends on the clinical scenario.
I would like to do a little bit of a plug for our science session next Sunday. We'll have Dr. Patty Dixon and Dr. Rich Steet presenting on 2 society-funded projects directed at sorting out what variants of unknown significance in IDS could mean. So this is a particular conundrum for MPS II. There may be mutations that look like they should cause disease but don't. And there are others that we would think maybe not cause disease, but do. So a clear prediction just based on sequence for MPS II is more difficult than in some of the other MPS disorders. So we've supported some of the science to help find a roadmap to a better approach towards discriminating disease alleles and non-disease alleles. So tune in next Sunday.
Okay, great. Thank you, Matthew. And I just want to take this chance to thank our speakers, Dr. Burton, Dr. Ellinwood, and Mr. Simon, for your great presentations today. I want to remind our audience that this is the end of today's sessions, and we will start tomorrow at noon Eastern with more sessions. A little bit about how the sessions, they are being recorded, but they will not be available immediately. It's available after about 48 hours, and then they're available for about a 60-day period. So put that on your calendar to go back if you need to look at sessions either you don't see or you want to see again. And the other thing is just to point out that the Society is committed to this newborn screening. We've carried the applications for 1 and 2 already. We've got the others in the process. 7 is probably maybe next, and then 4 and 6. But we're committed to carrying these and to the benefit of all of our kids and all our future families. So thank you again, and we'll sign off for today.
Thank you all.
Thank you.